The overall goal of this indirect immunofluorescent assay is to optimize the localization and quantification of endogenous and exogenous centromere-Kinetochore proteins, including centromere protein A and flag-tagged exogenous CNPA proteins in human cells.
This method can help answer key questions about how to identify and quantify centromere-Kinetochore proteins.
The main advantage of this technique is that it can be used to quantify different levels of centromere-Kinetochore protein expression depending on the selected antibodies of interest.
"18 hours after seeding, wash the cultured cells one time with PBS and add 500 microliters of reduced serum medium to each well of the 6-well polystyrene culture plate. Next, transfect the cells in each well with freshly prepared A and B mixture solution and incubate the cultures at 37 degrees Celsius and 5% carbon dioxide. After 4.5 hours, change the medium to high glucose DMEM supplemented with FBS and antibiotics and return the plate to the cell culture incubator. 48 to 72 hours later, aspirate the cell culture medium and rinse the cells one time with PBS, adding the saline down the sides of the culture wells to avoid disturbing the cells. Then fix the cells with 4% paraformaldehyde for 30 minutes at 4 degrees Celsius. At the end of the fixation period, rinse the cells 2 times with Kb2 buffer and permeabilize the samples in Kb1 buffer for 30 minutes at room temperature. Then rinse the cells one